My name is Kim Armstrong, and I'm a restoration biologist with River Partners. I'm going to be presenting on our Monarch project, Creating Habitat, a statewide experiment for the Western monarch. River Partners has partnered with the Xerces Society and Washington State University to enhance over 600 acres of monarch habitat statewide. We have 8 project sites ranging from San Diego in Southern California to Oroville in Northern California. Planting began in June of 2020, and the last site was planted in November of 2021. Some of our goals for this project are to make them easily repeatable, not just for us, but for other organizations. We're also interested in studying monarch egg deposition preferences. This includes pollinator plant plot size, presence or absence of vertical structure or shade, and the distance to water. We're also hoping to provide flowering resources from early spring to late fall, and to increase the diversity and abundance of pollinators at each site. Of the 8 sites, we have 3 location site types. The first type is enhancement of existing restoration sites, and that can be seen in the turquoise color on the map. The second site type is California Department of Fish and Wildlife property with flood irrigation, and that can be found in purple, and CDFW property without irrigation, and that is found in yellow. Of the project sites where it was possible, a plot-based experimental design was used, and these are the guidelines. The treatments. There were 2 different plot sizes, large and small, that were used on these sites. The large plots were 100 meters by 100 meters, while the small plots were 50 meters by 10 meters. And the conditions were based on available site conditions that included distance from water and vertical structure. The planting. The density of milkweed remained constant at 2,989 plants per hectare, regardless if it was a small plot or a large plot. At sites with irrigation, plugs were used, and at sites where there was no irrigation, rhizomes were used. Seed mixes were dependent on the site location, with flowering phenology from early spring to late fall. Some species included in the mix included California poppy, gum plant, lupin, yarrow, and lacy physalia. Here's an example of a plot-based design. This is the Feather River Wildlife Area, the Abbott Lake Unit. So we have 2 large plots in yellow and blue squares and 10 small plots in yellow and blue triangles. The plots were placed including or excluding vertical structure. The blue is close to shade while the yellow is in full sun. Small plots were 50 meters apart while large plots were over 250 meters apart. The distance to water was not included as a factor at this location. Monitoring on these sites included site visits from the Xerces Society. These took place 3 times per year between May and September and follows their streamlined bee monitoring protocol. She also recorded all pollinators that were in her transects at each site. Relevaes were done at plot-based design sites, while non-plot-based design sites received a full census. The Xerces Society also took a look at the plants for presence of monarchs. And this is an example of one of the 3 caterpillars that was found at the Hollenbeck Canyon Wildlife Area site. Some of the challenges we have faced so far have been sourcing milkweed, specifically ecotypes for our projects, sourcing pollinator forb mix seed, and the drought. But some successes we have had include the milkweed that is on the irrigation is doing really well, and we have monarchs on our project. And that concludes my presentation. I hope you all enjoy, and I look forward to answering your questions during the session. Thank you.